This slide is showing light and optics. So the first thing that we need to talk about is wavelength and color. Are the photoreceptors that are in our retina, they respond to wavelengths that are between 400 nanometers and 700 nanometers. So the eyes respond only to that visible light spectrum. There's an electromagnetic spectrum, which is much larger for things like UV light, radiation, microwave, but it's only with between 40 and 700, which is visible light. So when visible light passes through a spectrum, it's broken up into those bands of colors, the rainbow. You've probably seen that experiment when you were younger, maybe in a middle school class. Red wavelengths are the longest and have the lowest energy, whereas violet is the shortest and has the most energy. You may remember the mnemonic Roy G. Biv, which was red, orange, yellow, blue, indigo, violet. And when we talk about specific wavelengths, there is actually only going to be three very specific visual pigments that the cones respond to. And those are the blue cones, which is 420 nanometers, green cones, which are 530 nanometers, and then the longest wavelengths, which are the red cones at 560 nanometers. And then other colors that we perceive are combinations of different wavelengths. The next topic on this slide is refraction and lenses. And the word refraction means that it's the bending of the light rays. So the lenses refract or bend light because it's curved on both sides. It's convex, which means it's thicker in the center than at the edges, and concave means that it's thicker at the edges in the center. So the lens is what we call biconvex because it's going to be thicker in the center than at the edges. Concave would kind of be the opposite shape. It would look sort of like what I have drawn here, where it's now thicker at the edges than at the center. So the convex lens bends the light as it's passing through it, so the rays converge at the focal point. So the image formed at the focal point is essentially upside down and reversed from left to right. And we'll see this on the next slide. A concave lens does the opposite, so it disperses light, and this prevents light from being focused. So when we use a concave lens, it could be used for corrective vision. So if you look very closely at some lenses, you'll see that they are concave because they disperse the light based on what sort of abnormality the eye may have. We'll see that in a few slides. So when we talk about the refraction that actually occurs, let's first look at the pathway of light, which I mentioned earlier. So as light enters the eye, it first goes through the cornea, the aqueous humor, lens, vitreous humor, and then the entire neural layer of the retina, which is the photoreceptors. So you should know this pathway where the light travels through the eye: cornea, aqueous humor, lens, vitreous humor, and then finally the retina. So the light is refracted three times along its path, entering the cornea, entering the lens, and leaving the lens. So these are the only three places that should refract or bend the light. So the majority of the refractory power is actually in the cornea. However, that should be constant and shouldn't change focus, although that can be a problem in things like astigmatism. So what we see now is that the image is going to be inverted, upside down, and actually reversed. So imagine this is what it looks like, but this is how it's perceived in the retina. So the lens is what actually does the focusing, the fine focusing, not only for distant vision, but for close vision. So focusing on the focusing light on the retina looks like what we see here. For distant vision, that's actually what the eyes are best adapted for. So a normal eye means that you are 20/20, and that means that you have normal vision 20 feet away. So your eyes are sort of preset for distant vision. So there's no change that has to occur in lens shape for focusing. The cornea and the light focus light precisely on the retina. In this case, the ciliary muscles are relaxed in distant vision, and the lens is stretched flat. So the ciliary muscle and ciliary zone, you will focus an image by changing the shape. So the ciliary muscle is relaxed, and it stretches the lens basically flat in this situation. So the lens flattens for distant vision. And specifically, sympathetic input relaxes ciliary muscles. Because with sympathetic vision, you want to focus on what's in the distance. So we see a relaxed ciliary muscle and a flattened lens. For close vision, there's some accommodation that has to happen. So that accommodation basically means that the lens shape changes to increase this refraction, and we can see a near point of vision, and that's the closest point on which the eye can focus. So if you hold up a piece of paper to your eyes, and if you were to hold it real close, and then you had a lab partner, for example, measure the distance from your eye to that piece of paper, that would be the near point of vision. And obviously that changes as we age. You'll notice an older person is going to hold a piece of paper or the newspaper or an iPad nowadays further and further in the distance to read something. So that's a loss of accommodation that occurs. It's usually over the age of 50, sometimes it's earlier, and it's called presbyopia. So normal eyes would be metropia. Presbyopia is a loss of accommodation. And the last thing that has to happen for close vision is constriction of the pupils, and that prevents divergent light rays from entering the eye. So the things you need to remember is that the lens bulges for close vision, whereas it's flat for near vision. The parasympathetic division is activated, and that contracts the ciliary muscle, whereas the ciliary muscle is relaxed for distant vision. So some of the problems of refraction that we see are myopia, which is nearsightedness, hyperopia, which is farsightedness, and astigmatism. So this image shows you a normal eye. Myopia, nearsightedness, means basically the eyeball is too long. So what you'll see here is that the focal point would be in front of the retina at this point where this red line is drawn. So there needs to be a corrected lens for that. That corrected lens is going to be a concave lens, because that concave lens is going to diverge the light rays so that now they're going to touch the retina, come in contact with the retina, right where the macula lutea is. The opposite of that would be hyperopia, which is farsightedness, so the eyeball is too short. You can see what happens there is that the focal plane is behind the retina. So to correct that, a convex lens would be used. So if you looked really closely at the lens in the glasses, they would look like this here. So you should know the name of the imbalance and what type of corrective lens is used. The last imbalance on this slide is astigmatism. And astigmatism is unequal curvatures in different parts of the cornea or the lens. And that could be corrected with laser procedures like LASIK, for example. Now, there's a couple of adaptations that your eyes are responsible for: light adaptation and dark adaptation. And you don't need to know the exact chemical changes in your book, but essentially there's a chemical called rhodopsin, and it gets bleached even in starlight. So it's a chemical used. Bleaching occurs really fast. It's the chemical transformation, again, that you don't need to know, but it goes on in cones and in rods. So, as you know, the cones respond to bright light, the rods respond to dark light. Light adaptation would be moving from the darkness into bright light. So imagine coming out of a movie theater after a matinee. What's going to happen is the visual acuity is going to improve in about 5 to 10 minutes. The rod system turns off and the retinal sensitivity decreases. So the cones and neurons rapidly adapt. Whereas dark adaptation is exactly the opposite, moving from daylight into darkness. And if you imagine when that happens, the pupils dilate, the bright light bleached the rod pigments, so they're still turned off, but they need to adapt. And so that sensitivity increases within about 20 to 30 minutes. So when you go into a dark area, you really can't see much right away, but after a while, you can. So the visual pathway to the brain begins with these axons of the retinal ganglion cells from the optic nerve, and those medial fibers from each eye cross over at the optic chiasma, and the optic chiasma is where it becomes the optic tract. So there is decussation here where it crosses over to the other side. So if we look at this medial tract, it's now the optic tract back here, and that goes through the thalamus. Specifically, it's this nucleus. You don't have to know the name of it, but it forms the optic radiation and finally goes to the primary visual cortex where conscious perception of the visual image occurs.